Bacterial growth curve. The growth of microorganisms reproducing by binary fission can be plotted as a logarithm of the number of viable cells versus the incubation time. The resulting curve has four distinct phases: lag phase, exponential or log phase, stationary phase, and death phase. In addition, there are two more phases: acceleration phase and deceleration phase. Lag phase. When microorganisms are introduced into fresh culture medium, usually no immediate increase in cell number occurs, so this period is called the lag phase. Although cell division does not take place right away, and there is no net increase in mass, the cell is synthesizing new components. Physiologically, they are very active. A lag phase prior to the start of cell division can be necessary for a variety of reasons. The cells may be old and depleted of ATP, essential cofactors, and ribosomes. These must be synthesized before growth can begin. The medium may be different from the one the microorganism was growing in previously. New enzymes would be needed to use different nutrients. Possibly the microorganisms have been injured and require time to recover. Whatever the causes, eventually the cells retool, replicate their DNA, begin to increase in mass, and finally divide. The lag phase varies considerably in length with the condition of the microorganisms and the nature of the medium. This phase may be quite long if the inoculum is from an old culture or one that has been refrigerated. Inoculation of a culture into a chemically different medium also results in a longer lag phase. On the other hand, when a young, vigorously growing exponential phase culture is transferred to fresh medium of the same composition, the lag phase will be short or absent. Acceleration phase. At the end of lag phase, each organism divides, but all the organisms do not complete the lag phase simultaneously. There is a gradual increase in population until end of this period, when all cells are capable of dividing at regular intervals. This phase of gradual increase in population is called acceleration phase. Exponential phase or logarithmic phase. During the exponential or log phase, microorganisms are growing and dividing at the maximal rate possible, given their genetic potential, the nature of the medium, and the conditions under which they are growing. During this period, the cells divide steadily at a constant rate, and the log of the number of cells plotted against time results in a straight line. Their rate of growth is constant during the exponential phase, that is, the microorganisms are dividing and doubling in number at regular intervals. Because each individual divides at a slightly different moment, the growth curve rises smoothly rather than in discrete jumps. The population is most uniform in terms of chemical and physiological properties during this phase, therefore, exponential phase cultures are usually used in biochemical and physiological studies. Exponential growth is balanced growth. That is, all cellular constituents are manufactured at constant rates relative to each other. The time required for the population to double is called the generation time. Number of generations per hour is called growth rate, and it is reciprocal of generation time. Deceleration phase. Finally, the growth may cease when a critical population level is reached. There is a gradual decrease in population at the end of this period when the cells start to die. This phase of gradual decrease in population is called deceleration phase. Death phase or phase of decline. Nutrient deprivation and buildup of toxic wastes lead to decline in the number of viable cells characteristic of death phase. The bacteria may die faster than new cells are produced. The death of microbial population is also logarithmic. Death is mainly due to depletion of nutrients and accumulation of inhibitory products such as acids. Death is defined to be the irreversible loss of the ability of microorganisms to reproduce.